Thank you for having me. It's awesome to be here. I'm here today and I'm going to introduce you to our HFSP-funded project, Micromaze. So it's a real honor to represent my collaborative team by talking to you today. So what we are interested in is species coexistence. This is a big concept and a big problem in ecology in terms of our understanding of this phenomenon. So species coexistence happens across our globe, from temperate forests to rainforests to arid and desert ecosystems, and even in aquatic and marine systems as well. What we are seeing here are species coexisting and living together peacefully within their environments. Why is this? Why do we see species coexistence rather than monocultures of species dominating the surface of our planet? Why is it that we find multiple species of plants living together, of birds, of fish, of insects, rather than just one or two completely monopolizing the resources of our planet? This is a fundamental question in ecological science. And traditionally, our understanding and our explanation of species coexistence has relied on the idea of niche differentiation. And we can think about this as I'll describe now. So typically we think of species as having niches. They need particular food resources or nesting areas or certain environments that they can live in. And what we tend to think is that if species overlap strongly in their niches, then they will outcompete one or the other, or they will compete and the stronger competitor will outlast the weaker competitor. So in this example here, we've got a rabbit and a squirrel species, but they eat different things and they nest in different places. So they can peacefully coexist. Their niches are not overlapping. But if we introduce a third species into this mix that eats the same food as, as our squirrel, we might expect that the squirrel and the mouse would compete for this food resource and one or the other would go locally extinct and coexistence would not happen. In this case, our mouse lives in a different area to the squirrel, so their niches aren't overlapping strongly enough and they can therefore coexist. But our ideas about coexistence all stem from this idea of niche differentiation. The problem is that these ideas don't always work. We can look at many examples across our planet. Two here are the African Great Lake cichlid radiation and Australian savanna ants. In both of these cases, we see tens or hundreds, in the case of the ants, of species coexisting together in a relatively small space, 100 by 100 meters in area for the ants. And there are some niche differences. Some of these species eat different things, some of the fish live at different points in the lake, but there are not enough niches to explain why there are so many species that all coexist together. So this niche explanation doesn't fully solve this problem of coexistence. Something else must be going on. And what we think on our HFSP consortium is that a concept called the microclimate may help us to understand coexistence. And I'll explain this by first talking about the macroclimate. The macroclimate is what we all experience today walking around Geneva. We typically measure the macroclimate from weather stations like that pictured on the slide. Stevenson screens. These are devices set 2 meters above the ground and they measure temperature, wind speed, rainfall, humidity levels, etc. And what we can do is link up networks of weather stations to interpolate global climatic patterns. And you can see an example of this on the bottom left there. And these are typically gridded at relatively coarse scales, multiple kilometers squared, for instance. The problem is that most organisms do not experience the macroclimate. Most organisms are living in the microclimate. So consider this ant on the top right of the screen. She's not experiencing the temperatures that we do 2 meters above the ground. 2 meters above the ground up here. Not 2 meters. She's living on the ground surface. And for her, the temperatures and the conditions she experiences are strongly influenced by fine-scale vegetation, topographic patterns and conduction and convection forces. She's not experiencing temperatures that our weather stations are recording. So at the microclimate scale, these climates are much more variable and much more extreme through space and time than what we would estimate from our weather stations, from our macroclimatic measures. And just to emphasize this further, just above me on the bottom right here is a thermal image of a 1 by 1 meter area of rainforest. So this is capturing the temperatures in a very small area. And here we see that temperatures are varying, and they're finely structured as well, from around 27 degrees up to 44 degrees Celsius. So that's this huge temperature variation in a very small space. So these kinds of extremes in microclimatic conditions are what most organisms and animals on our planet live within, rather than these broad-scale averaged macroclimatic patterns. So how does the microclimate fit in with this idea of species coexistence? Well, this conceptual diagram sort of sits at the heart of our project. And you can imagine this a little bit like that thermal image I just showed. Each pixel might represent the temperatures or some other environmental variable at a given spatial and temporal point. And our idea with this diagram is that these black walls of this maze-like structure represent intolerable microclimates, conditions that are too hot, too cold, too wet, too dry for a given organism to traverse across. And then these spaces in between might represent tolerable and comfortable conditions. These different colors then may represent species that all otherwise seem to have the same niche. But in this system, they are spatially segregated from each other. They're effectively isolated from each other by this fine-scale maze-like structure of the microclimate at a very fine scale. So if these species with similar niches don't actually meet each other that often in practice, they don't therefore compete, and coexistence can be promoted. So our idea, our hypothesis, this microclimate maze hypothesis, is that this fine-scale microclimate structuring may promote coexistence by effectively isolating species that would otherwise compete. And we're trying to investigate this idea by focusing on mountains and ants. So we're working in the Cederberg Mountains in South Africa. These are about 3 hours north of Cape Town, 3 hours drive north of Cape Town. And they are dominated by a Mediterranean vegetation type called the fynbos. This is a highly biodiverse and endemic vegetation type full of protea plants that you see pictured on the top right. So it's a beautiful and very exciting landscape to work within. But there are also climatic extremes at both the macro and the microclimate level throughout this ecosystem, linked with elevational changes and also strong seasonal swings. And we're then investigating the coexistence patterns of ants. Ants are a globally important insect group. There's over 15,000 species and are critical for many ecosystem functions. But for our purposes here, what's really interesting is that there's a few hundred species living in this region, but at a very local scale, in an area smaller than this room, we will find upwards of 40 species seemingly coexisting together. And what's important about ants is that they are social organisms. So all the individuals you see on these pictures are sterile, female, non-reproductive worker individuals. What they do is effectively act like mobile leaves or mobile roots, if we can draw an analogy with plants. They leave their central nest, they forage throughout this dynamic, variable microclimate and bring resources back to their central nesting location. So this makes them an ideal model system for testing our microclimate maze hypothesis. So our project is split into four sub-projects, as it were. And each of them has some specific hypotheses, and they're all coming together to tackle this microclimate maze idea. So our first component is about estimating the empirical microclimates across the Cederberg and within the fynbos vegetation system. And we're measuring these using fine-scale sensors deployed across the landscapes that we're working within. Secondly, we are then measuring the topographic and vegetation structure of these landscapes. And we're doing that using LiDAR hooked up onto drones. So we're flying drones with LiDAR systems attached to them across our sites, and these are effectively 3D scanning the environments. This is giving us a 3D model of what the vegetation and the mountains and fine-scale topographic variation looks like. We can then combine our empirical estimates with these 3D models to produce four-dimensional, through space and time, maps of what the microclimate looks like from the perspective of a small organism like an ant. On the other side of our project, we are estimating the fundamental niches of the ant species. We're doing this in many ways, but the most important one is how ants respond to temperature and humidity conditions. So we're running a range of lab assays to estimate these kinds of niche parameters from the 40-plus species that we're working with. And finally, we are then quantifying the realized niches of these species. So in the lab, we measure what's possible, and out in the field, we measure the conditions that ants are actually foraging under. We're also then recording where these species live in the fynbos, where are they positioned in this maze, how far they forage, and what kinds of food and other items are they bringing back to their nests. We're then combining all these data streams in a synthesis project which will ask our big microclimate maze hypothesis questions. What I'll do now is just show you some of the data outputs and some of the initial results that we're getting from our work here. We're about halfway through our project. So this diagram here is showing you the outputs of our LiDAR scanning. So in the middle here, we've got one of the four landscapes we're working in, and those four little red plots are these focal plots that we use to study in fine scale the ant physiology and behavior. And this green color is representing an index called Plant Area Index. Lighter green means there's less plant material, darker green means there's more. So this is giving us a very fine scale map of where plant structures are within this landscape. What we then do is parameterize a biophysical model with our empirically measured microclimate data to transform this into an estimate of what the microclimate looks like at a very fine scale, but at a relatively large extent. So this is the same landscape, and these blue to red colors are now representing average daily temperatures across the year, blue being cold and red being hot. So we have this for every hour of the year that we've been modeling across all the locations that we've scanned. So you can already see here big differences in what the average temperatures are, dependent on whether you're in a valley, near a stream, or towards the crests of the hills. And even within our focal plots, which are 50 by 50 meters, there is large differences in the average temperatures, largely driven by patterns of vegetation, which can cause shade, and small-scale topographic differences. And our initial results are hinting that this microclimate maze hypothesis might be true. So what this data set here is showing you is the number of ant species on the y-axis. We measure this by using an old-school technique called baiting, which you see on the left. So you put out food for ants, and we count who comes. And on the x-axis, we're measuring thermal heterogeneity. How variable are those plots that we're working in terms of temperature? And here, if you squint a little bit and trust our statistics, we see a positive relationship between thermal variability and the number of species that we find, which fits in with the predictions of the microclimate maze hypothesis. This doesn't test it explicitly. So what we are working on now is to collect a range of data sets that will allow us to explicitly investigate this microclimate maze hypothesis. So these data here are showing us a number of species on the upright axis and the surface temperature at which these species forage on the x-axis. So what you see here are some heat specialists at the very top. We see some cold specialists towards the bottom. But in the middle, you can see that most of our ant species are foraging at around 20 to 25 degrees Celsius on the ground. So there's strong thermal niche overlap amongst most of these different species. So these data represent the temperatures at which these species are actually active and moving around their microclimates. What we're also recording are the distances that different species will forage. So on the upright, we've got species again, and on the x-axis, we're measuring foraging distance. This is a log scale here. So some species forage a few centimeters to a meter perhaps from their nest, and others towards the top will be foraging up to 30 meters from their home base, their colony. What we are then going to do with these data is put them in a spatially explicit context. So you imagine we might have a single plot where microclimate is varying dynamically through time. We can then pinpoint exactly where different species live in this system and understand what their foraging extent is. How far might they forage? And therefore, what is the microclimate that they will experience and encounter during the course of a day or the course of an entire year? Our plan is to then overlay these different kinds of microclimate maze maps for all our different species to understand how they overlap and therefore how coexistence might be maintained. I'll wrap up there, but thank you very much for listening. A big thank you to HFSP for having us here and for awarding us this grant. It's been a great pleasure and honor to work on it. And a thank you to all my collaborators, some of whom are in the room, and our various institutions for supporting our work. Thanks for listening.